Hello students, today we are going to discuss about two important topics, and that is polygenic inheritance and pleiotropism. So in previous lecture we have already discussed that some theories are there that do not follow Mendel's law. Okay, in which you can see incomplete dominance, okay, here codominance and multiple allelism. These three theories which already we have discussed in previous lecture, and these all are considered as a non-Mendelian inheritance. Okay, we are going to learn about two more theories about this, okay, and that is polygenic inheritance and pleiotropism. So today in today's lecture, we are going to discuss about these two theories, polygenic inheritance and pleiotropism. These two also do not follow Mendel's law, okay, and that's why they are calling as a non-Mendelian inheritance. So let me start with the polygenic inheritance. Okay, so what is a polygenic inheritance? Poly means more than two. Okay, genics means more than two genes are responsible for inheritance. Okay, so let me see. It is the inheritance in which some traits are controlled by several genes, more than two genes. Okay, that's why it is called multiple genes. Okay, that's why name is given polygenic inheritance. Okay, so let me discuss more with the example. Here one example is there, and that is human skin color. Even human height is also one example of the polygenic inheritance. Okay, so human skin color, you know very well, different shades of skin is there on the earth. Okay, different shades are there, and that is just due to polygenic inheritance. And you know very well, height difference is also there in so many people. You can see different height is there in the human. So these both the character, human skin color and human height, both are one type of a polygenic inheritance. Okay, so let me discuss that. What is a polygenic inheritance? It considered the influence of environment. Okay, and in a polygenic trait, what happen? Phenotypic reflect means phenotypic character will reflect the contribution of each allele. Okay, so here phenotypic reflect the contribution of each allele, means each allele will responsible for the phenotypic reflection. Okay, for example, the effect of each allele is additive. So you must be remember, whenever allele is added, then character will also added. Okay, more number of genes which is responsible for skin color. If more number of genes are added, then skin color will be darker side. Okay, and less number of genes are having a lighter skin color by this way. Okay, so here effect of each allele is additive for the any character. Okay, so let me discuss more. So human skin color is an example of a polygenic inheritance. Okay, so here let me discuss about this. Assume that three genes. Suppose here you can just assume that three genes, capital A, B and C, these three capital will control human skin color. Just you have to assume. Okay, dominant form is A, B and C capital, responsible for dark skin color. You must be remember these all three genes responsible for dark color. Okay, and recessive form is small a, small b, small c. These three recessive form are responsible for light skin color. So you must be remember here, just you have to assume, okay, a, b, c. If it is capital, then skin color will be dark. Okay, and if small letter recessive form is present in the skin, okay, then light skin color will be there. So let me discuss more. Okay, so genotype with all dominant allele, just this is the example. Okay, genotype with all dominant allele, here you can see all are dominant. Okay, capital A, B, B, C, C. This is the darkest skin color. You must be remember, this is the darkest skin color because all character, all alleles which is present in the person having dominant allele. Okay, whereas this one, here you can see if genotype with all this one, this you can see all recessive, all small, then it will give lightest skin color. So in the presence of how many number of genes dominant present in the skin color, how many number of recessive gene presence in the skin color will define skin color tone. Okay, so genotype with 3 dominant or 3 recessive will give intermediate skin color. So you have to remember here, these or these will define the skin color tone. Okay, so if more number of these capital is present, then it is responsible for dark skin. Okay, more number of these small is present, then it will be responsible for lighter skin. And if intermediate is present, suppose one example I have taken, there is a capital A, small a, capital B, small b, capital C, small c, means half is a recessive and half is a dominant, right? Out of this 6, half is a dominant and half is a recessive. Then what happen? Intermediate skin color will be observed. So this is the case. Okay, so you must be remember, more number of dominant allele if present, it will give dark color. Okay, and more number of recessive if present, then it will give lighter color. Let me give one more example. Suppose here 2 dominant is there. Okay, 2 dominant is there. Okay, and then 4 recessive is there. 4 recessive is there. Then what happen? There is a skin color having more lighter shade. Okay, and if more dominant is there, then this allele is less present, then what happen? This darker shade will be dominant. So by this way we can get the idea which color skin shade will be there. Thus, the number of each type of allele in the genotype determine the darkness of the or lightness of the skin color. Okay, it is all depend on the number of allele present in the skin. Okay, so which type of alleles are present, whether it is more darker side or a lighter side, it will depend on the presence of these all allele. Okay, so this genotype will determine the darkness or lightness of the skin color. So these all are the example of a polygenic inheritance. So now let me try to understand by using this Punnett square analysis. Okay, so here you can see a sperm possibilities. Okay, and here you can see eggs possibilities. Okay, and here you can see this is a white color spot and dark black color spot. So just you have to assume that this light color is indicating a recessive alleles. Okay, small a, b and c. Okay, and here dark color spot indicating capital letters, capital A, capital B and capital C. So here in first box you can see if these two alleles are mixed together, here sperms and this one is the egg, if fused together it will result in the light color offspring, right, because of absence of this black color dot. Okay, so this means here only recessive gene will be merged. But here what happened? One, you can see one here, this one dark color is indicated. Okay, that's why here what happened? It will be darker than this one. This will be darker than this one. Okay, and if 2 is added, then this will be darker than this one. Okay, so out of these 1, 2 and 3, which one will be darker? This one will be darker. Okay, this will be intermediate and this will be lighter, like that. And if more than 2 is added, more than 2, here you can see 3 black is there. Okay, then at that time what happened? The shade of these, suppose this is the A, B, C and this is the D. Okay, so A, B, C and D. So here D will be darkest. This one will be darkest. Okay, and these will be intermediate. This will be lower than this one and this one be lighter than this. So by this way you can get the idea how many capital letters are present or how many dominant alleles are present in the organism. It will define the darker shade. Okay, and whatever the number which is the recessive one is present, which will define the lighter shade. Okay, these all are the possibilities of a skin color. Okay, and here you can see this one. If you compare this and this, okay, if you compare this one with this, suppose this is A and this is B. Okay, which one will be darker? So this one will be darker than this. Okay, how? So here only 3 this dominant gene are present, dominant allele are present, but here you can see 4 dominant is present. Okay, so it is depend on the how much amount of a dominant gene is present or allele is present in the person. It will define darker shade. Okay, and this will be darker than this one. Okay, and if you compare these three, here you can see 1, 2 and 3. This is the A, B and C. If you compare in these 3, then what happened? This is the lighter just because of 2. Okay, and this is the intermediate because of 3 and here 4 is there. So out of these 3, if you compare these 3, then you will observe this is the darker. Okay, this will be intermediate and this will be lighter. So by this way you can compare that. Okay, and here in this case, these two, if you compare these two, okay, then what happened? This one will be darkest. Okay, because here you can see all alleles which is present, all 6 alleles which is present in the organism, all are capital. Okay, and here you can see one is a lighter recessive allele is present, and that's why a skin color tone of this organism will be lighter than this one. Lighter than this one. But if you compare this organism with this one, then what happened? This will be darker than this one. Okay, so it is all possible. You can see the combinations. Okay, so it will define the skin color. So you may get the idea which color, skin color tone will be there on the presence of alleles. Okay, so if alleles which are all are recessive alleles are present, then skin color will be light, and if all are this dominant is present, the skin color will be darkest. Okay, so you must be remember this all. So here in this slide you can see 5 uncle photograph is given. Okay, uncle A, this one, all are bald uncles. Okay, so uncle A, uncle B, uncle C, uncle D and E. But due to some problem, what happened? Their genotype is exchanged. Okay, due to some problem, what happened? Genotype was exchanged. Okay, here you can see uncle A, B, C, D and E. Okay, but here you can see genotype is not matched with their skin color, right? Just you have to match it out. Okay, so if uncle A is there, which will be the genotype of this uncle A out of this, because all genotype which is given here is interchange. Okay, exchange and interchange, and that's why just you have to identify what will be the genotype of uncle A, what will be the genotype of uncle B, and what will be the genotype of C or D or E. Okay, out of these all P, Q, R, S, T. Okay, and for that I am giving you 2 minutes. Okay, your time start now. Okay, your time is over. Just write down your possible answer in a comment. Next theory is a Pleiotropism, and this is an exceptional case, okay, that do not follow Mendel's law, okay, and that's why we are calling it as a non-Mendelian inheritance. Okay, so what happened here? According to Mendel, you know very well, one gene is responsible for one phenotypic expression, right? But here what happened? Only one gene will exhibit multiple phenotypic expression. That means only one gene will responsible for more than two type of traits or expression. Okay, and such a phenomena is called pleiotropism. And such a gene which express like this is called pleiotropic gene. Okay, so let me repeat. A single gene exhibit multiple phenotypic expressions. Such a gene is called pleiotropic gene. Okay, in most cases, mechanism of pleiotropism is just due to effect of a gene on a metabolic pathway. Okay, we will learn in detail in next lectures. Okay, with a contribute towards the different phenotype, and this we will learn in detail in next lecture. And such an example which shows pleiotropism is a star synthesis in the pea plant. Okay, phenylketonuria. These diseases we will learn in detail in this chapter. Okay, in next lectures. Sickle cell anemia also we will learn in detail in this chapter in next lectures. Okay, so what is a sickle cell anemia and what is a phenylketonuria? We will learn in detail. So here the case of a phenylketonuria, and this is a sickle cell anemia. Okay, in phenylketonuria and the sickle cell anemia, the mutant gene. What is a mutant gene? Which we will also learn in next lectures. Okay, so mutant gene is a one lecture, full lecture we will learn in this. Okay, so mutant gene has many phenotypic effect. Okay, that is a modified gene, and that's why it is called mutant gene. We will learn in this detail in next lectures. Mutant gene has a many phenotypic effects. Okay, for example, phenylketonuria cause mental retardedness also. Suppose one gene is mutated, then it will also cause mental retardedness, reduction in hair, skin pigmentation, like that. So you must be remember what will be done Okay, in case of a pleiotropism. So here if a phenylketonuria related gene is there, then what happen? If it is the condition, then mental retardedness also occur, reduction in hair, skin pigmentation. Means one gene is responsible for more than 2 or 3 characters, okay, or a trait. So mental retardedness along with the reduction in hair, skin pigmentation all will occur just because of this phenylketonuria related gene or a mutation. Okay. So this is your homework for today's lecture, and before we finish this lecture, let me summarize whole topic by using one animation video. So look at this video.
What do snapdragons, human height, and speckled chickens have in common? Well, they are easily identified as non-Mendelian traits. By non-Mendelian, we mean that genetically, they are rule breakers. They don't follow the regular Mendelian rule that having a dominant allele means the dominant trait will show. Remember in our guinea pig video, having a dominant allele represented by a capital H meant that the guinea pig would have hair? Only if there was no dominant present, a genotype of little h, would there be a hairless guinea pig. Well, that's a Mendelian trait. But what we're going to cover now is when these traits are non-Mendelian and they don't follow these basic rules. So let's first start by snapdragons. We confess that when we heard this word, we thought they were some really amazing kind of creature. Well, they are amazing, but they're flowers, so I don't know, that's not exactly what we envisioned. Well, in Snapdragon Genetics, there can be 3 phenotypes: red, white, or something in between, pink. It's called incomplete dominance. In incomplete dominance, the dominant allele is not completely expressed when the recessive allele is around. There isn't really a clear dominant allele. If you cross a red flower, and we're going to write it here big R, and a white flower, little r, you are going to get offspring that are big R, little r. But unlike a Mendelian trait, if this is incomplete dominance, that big R allele is not completely expressed when the little r is around. So big R, little r in this case is pink. If you cross 2 pink flowers, big R, little r, like shown in this Punnett square here, you can get offspring that are red, white, or pink. Incomplete dominance is different from codominance. Codominance, like a co-worker, that prefix co should make you think together. They work together, the alleles, that is. For that reason, we like to use different letters entirely to represent the alleles. There is a codominance involving color in some types of chickens. Take a look at this Punnett square. If you cross a black chicken, represented here by BB, and a white chicken, represented here by WW, all the offspring here are BW. BW chickens are both black and white. They're speckled. See, both traits show up. This is the essence of codominance. And you know, what's more awesome than a speckled chicken? Well, except for those silky bantam chickens. By the way, you will find that many people use different formatting for incomplete dominance and codominance. Some people like to use different letters entirely for incomplete dominance, and some people don't. Some people also prefer to do exponents with codominance, and some people don't. Formatting aside, when you're solving these Punnett squares, since we find this really does vary from classroom to classroom, the real concept is that in incomplete dominance, one allele is not completely dominant over the other, so you see this almost in-between phenotype. In codominance, both alleles are expressed. It's the application that matters here. Height is fascinating. In our immediate family, Pinky is taller than Petunia. Our mom is also taller than Petunia. So how does that happen? There isn't just one height gene. There's a lot of genes that determine your height. What I mean by that is that you don't just have a pair of alleles, like big A, that will code for your height. It's more like someone having a genotype of big A, big B, little b, big C, little c, and maybe more genes to ultimately determine your height. And you inherit one allele for each of the height genes from each parent. All of these genes work together to determine your height. Your skin color is also determined by many genes, just like your height. These are called polygenic traits. Poly means many, so many genes coding for one trait is what polygenic means. By the way, both height and skin color can be influenced by environmental factors as well. Nutrition growing up can affect your height, just as spending a lot of time in the sun can affect your skin color. However, this doesn't change the genetics for this trait. And finally, one more thing that we want to talk about. Epistasis. Epistasis is when one gene really depends on another gene for it to even be expressed. I mean, it really depends on this other gene. So let's pick an animal like, a llama. Let's say this llama has a dominant big B allele, which means its wool will be black. So big B, or big B, little b means that it will have black wool. And let's say that if a llama has a pair of recessive alleles, little b, then it will have brown wool. Now, what if there is another gene that controls whether this pigment will even be expressed in the llama in the first place? In this hypothetical example, a llama could have this other gene coded for by big C, big C, or little c in its genotype. However, if a llama has the genotype little c, it will not allow the other gene for wool color to even be expressed. Since we have two genes here, the gene for wool color and then this gene that controls whether the color is even expressed, this calls for our favorite 16-square dihybrid cross. If you notice in this dihybrid, crossing two heterozygote llamas, any llama with big B and big B will typically give a black llama, and little b will typically give a brown llama, in all cases unless they inherit a little c. If the llama has a little c in its genotype, then the gene for wool color is not expressed, and the llama is albino. This means that no pigment is expressed at all.
In next lecture, we will learn about chromosomal theory. So till that, goodbye.